All right, guys, let's talk hormonal regulation. Hormonal regulation is the basics of endocrine signaling, which we'll be looking at. We'll also look at some very specific types of hormonal regulation on metabolism, for example, insulin and its relation to things like obesity. So, in all of our previous lectures, we have focused on metabolism at a cellular level. We looked at where things were happening within the organelles of the cells, the roles and mechanisms of very specific enzymes, the flux of metabolites through pathways, feedback regulation on pathways themselves, how the pathways are regulated, and transport of metabolites across organelles in inside the cell. But in this chapter, when looking at hormones, we actually have to think about the whole organism level. And that's because hormones signal across organisms, across cells, not within the cell only. So, this includes the role and structure of specific tissues and organs, flux of metabolites from organ to organ, and hormonal control over all metabolism within an organism. And then, of course, relations and more specifically, control of body mass. Hormones are small molecules or proteins. They can be either of those. They can be small metabolites or small proteins that are produced in a tissue and released into the circulation of an organism, carried to another tissue where they then act and bring about cellular activities. This is the base definition. This can be in numerous types of organisms, not just humans. Many organisms have types of hormone. Um, and again, they can range in what they look like as far as small molecule to protein. They serve generally to coordinate metabolic activity of several tissues and organs. It's the way that uh, whole systems of cells in a tissue or in an organ actually talk to each other and relay information about metabolism. So, virtually every process in a complex organism is regulated by one or many hormones. This includes things like maintenance of blood pressure, blood volume, the electrolyte balance in your blood, sexual differentiation, development, reproduction, hunger, and your eating behavior, your digestion, and just your fuel allocation throughout the organism. Now, there are neuronal signaling versus hormonal signaling. In neuronal signaling, nerve cells release neurotransmitters that act on nearby cells. This is a similar process. You might think that neurotransmitters, you can confuse them with hormones. Again, this happens specifically when you go from a nerve cell to another cell. This can be from nerve to nerve. It can be between a nerve and muscle or nerve and other cells, for example, intestinal cells. The distance here is very small. The nerves, right, they reach all the way through down our spinal cord um, to these other cells. Um, and the actual release of the neurotransmitter, the distance it travels to get to the other cell is very small. Hormonal signaling occurs when you have molecules that are carried through the bloodstream to other cells or other organs, right? So the distance that hormones travel is very large. They can travel 1 meter or more, for example, versus neurotransmitters, which only travel micrometers. And the coordination of metabolism in a mammal is achieved through something called the neuroendocrine system. Hormones are usually produced in very small amounts, and it's usually difficult to purify them if you had to actually get some from blood. For example, chemical analysis of the thyroid releasing hormone, or TRH, from pigs requires 1 million hypothalamuses, or a million pigs, to do the accurate get an appreciable quantity, excuse me. Now, radioimmunoassays were developed to be more sensitive way to measure hormones instead of actually seeing an appreciable amount by taking out 1 million hypothalamuses. And also detection through ELISAs, which was a method that we looked at in Biochemistry 1. Remember, this uses antibodies to label hormones, or really anything. You can use antibodies to label any type of molecule. Mass spectrometry is also a way to assess hormones, because these machines can measure very small ranges or quantities of the hormones. So just a reminder of the ELISA method. This is where a purified hormone, you can see, is added. So we have our sample that coats the plate. So this is our sample with the antigen, our hormone, for example. And then you incubate in, after they are coated on the plate, a primary antibody that can bind only the hormone or antigen you are looking for. And then a secondary antibody is added, which only binds the primary antibody, but this secondary antibody has some type of component on it which allows for the changing of color. It can be colorimetric, or it can allow the molecule to fluoresce, for example. And it acts as like a beacon to show you how much of the antibody is present. Now, there are 3 types of hormones that we can classify, the major types. These are protein hormones, steroid hormones, and amine hormones. Protein hormones, or polypeptide hormones, are made of chains of amino acids. They are exactly what they sound like. They are small proteins, or polypeptides. One very good example of this is ADH, which is antidiuretic hormone, which decreases the blood pressure, our blood pressure. This is a small polypeptide hormone. There's also steroid hormones, which are derived from lipids. These are reproductive hormones such as testosterone and estrogen. These are good examples. There's also amine hormones, which are derived from amino acids. We saw these in our chapter about amino acid catabolism and anabolism, that some amino acids can be converted into things like epinephrine, which helps regulate our fight or flight response, and again is based on a amino acid structure. The hormone receptor interaction, so the point in which you have some receptor, right, binding some hormone. This is your hormone, and this is your receptor. This interaction is very specific, and they have to have very high affinity for each other. So different cells actually have different sets of receptors, and different cells with the same receptor can even have different downstream effects. Even structurally similar hormones can bind different receptors. These interactions are high affinity so that only low amounts of hormones are needed. So this is looking at downstream effects that can occur after a hormone interacts with a receptor. These are different types of downstream effects. They're very common downstream effects that lead to particular goals, but kind of the chain in which after the receptor is activated, the chain of activation, of downstream activation, can go through one of these 5 main events. One type of downstream event can utilize what's called a secondary messenger, which gets released inside the cell, and then that can regulate enzymes that need to be turned on or off. There are certain types of receptor tyrosine kinases that can be activated as a downstream effect. Another result is that a hormone-gated ion channel is opened or closed, and this changes the membrane potential of the cell, which will lead to certain effects. You can also have adhesion receptors that send information to the cytoskeleton and actually change how the cell is shaped or its rigidity. There's also steroid-bound receptor proteins in the nucleus that can alter gene expression. Now, hormones can be extracellular or they can be intracellular. Generally, peptide and amine hormones bind to the outside. They bind extracellularly. This includes things like insulin. It is a peptide hormone. Or epinephrine, which is an amine hormone. They bind to receptors that actually span through the membrane and ultimately induce a conformational change that produces a secondary messenger inside the cell, which allows for amplification of many targets. So here you can see we have our cell surface receptor. Here's the whole receptor, and it actually spans through the membrane. The hormone binds to one side, and that elicits a reaction on the other side of the receptor when it binds, releasing some secondary messengers and then sending signals off within the cell. This is opposed to something like a nuclear receptor, which usually uses a steroid or thyroid hormone, and these can just enter straight into the cell. As you can see, steroids like retinoid, vitamin D, and thyroid hormones are very non-polar, so they can go through the lipid membrane. These are intracellular type steroids that usually need to bind into the nucleus of the cell. So they will transfer all the way through the cell membrane and into the nucleus where they can bind to DNA and act as a transcription factor. This usually increases or decreases the expression of certain genes in a cell or in a tissue. And this process can take hours to even days to ultimately occur with the effect. Hormones can further be classified based on their path or from release to target, or excuse me, their path from release to their target. So this is just a different type of classification system that can be used that describes the path the hormones are taking. For example, paracrine are released into the extracellular space and they diffuse to neighboring targets. These are things like eicosanoids, which we looked at, are related to the inflammation processes that are targeted by NSAIDs. Endocrine are released into the blood, carried to target cells. These include things like insulin and glucagon. And then autocrine affect cells where they're produced, but they bind to a surface receptor. So paracrine, again, are going to be traveling shorter distances. They're usually diffused close by. Endocrine goes into the blood, and they are carried to target cells. And autocrine is where you are affecting the same cell that it's produced in. So here's some examples of those classifications. I'm talking about the type of steroid, or excuse me, type of hormone, and then the classification as well. So you can see something like insulin and glucagon. These are a peptide type of hormone. They bind in an exterior fashion to the membrane and use secondary messengers to signal. And they are a type of endocrine in which they are released into the blood and travel. So now we're going to look at some of the more important hormones in a little bit more detail. One of the first ones I just mentioned, and probably the most recognized hormone, is insulin. Insulin is a peptide type hormone, which is synthesized from a type of cell called the beta cell. And it is synthesized first as a molecule called pre-proinsulin. This then has to be actually processed into its active form to further do any signaling. It's secreted in this form in response to increased blood glucose levels, and ultimately it produces secondary messengers inside its target cells. It can bind to receptors in muscle, brain, liver, adipose tissue, and other fuel metabolizing tissues. These are generally the types of tissues that produce a receptor that will accept the insulin hormone. Here is the mechanism of actually making mature insulin. Again, mature insulin has to be formed from this pre-proinsulin, and it is processed through a series of proteolytic processing in which it is actually cleaved, parts of it are cleaved. There's a removal of 23 amino acids at the amino terminus, and formation of 3 disulfide bonds to actually give it some structure. You can see that mature insulin is actually made of 2 chains held together by these disulfide bonds. This is called the A chain and the B chain. Now let's look at another type of hormone, which is called the catecholamine hormones. These include epinephrine and norepinephrine, and these are synthesized in the adrenal glands. They're actually synthesized from amino acids, L-tyrosine, so they are an amino type of hormone. You can see catechol, which is kind of the one of catechol, epinephrine and norepinephrine, and you can see their similarities. They are similar in structure and derived ultimately from the same structural type. These hormones are concentrated in vesicles in certain cells, and then they are released. This is very similar to how peptide hormones are stored and then released. Again, these bind extracellularly to receptors and generate secondary messengers similar to peptide hormones. But remember, these are based on the amino acid-like structure. Epinephrine, once it binds to its receptor, it activates adenyl cyclase, which produces CAMP, that is cyclic adenine monophosphate. You can see epinephrine will bind up here to the receptor. This sends us a processing signal over to adenylate cyclase, which ultimately makes this cyclic AMP or CAMP. And CAMP further signals, this is another secondary signal, signals to activate protein kinase A. Protein kinase A can then activate glycogen phosphorylase B, which breaks down glycogen to glucose 1-phosphate for extra energy. Essentially, there are millions of copies of glucose 1-phosphate, and this process can take mere seconds. And that's because of this signaling process in which you're using secondary signal messengers to amplify the signal. So even though one epinephrine binds, it actually will then make many secondary messengers, which then can act on many copies of glycogen and make many copies of glucose 1-phosphate. Another main type of hormone, the eicosanoid hormones, these include prostaglandins, thromboxans, leukotrienes. These are all based on lipids. They come from phospholipids and originate from a base lipid called arachidonate. These are produced when needed from the arachidonate or arachidonic acid via a enzyme called phospholipase A2. These are just paracrine hormones. Again, that means that they act on nearby cells. They do not travel the bloodstream generally. So normally they're being released by a nearby target and then acting on the cells. So for example, in the case of inflammation, they play a role in inflammation and smooth muscle contraction. Usually the site in which you have some type of injury will produce these prostaglandins, and those then will signal to nearby cells to become inflamed. They are related to the mediation of pain and inflammation in most all tissues. Another type of lipid hormone is the steroid hormones. These include cortisol, testosterone, and estradiol. These are all produced from cholesterol. So they're all synthesized as lipids from cholesterol. And they can bind to carrier proteins and travel through the blood system. Now, these lipid type of hormones, remember, are generally intracellular. They tend to bind into the cell nucleus and then alter gene expression. So for example, glucocorticoids, which is a type of hormone like cortisol, they primarily affect metabolism of carbohydrates. Mineralocorticoids such as aldosterone regulate concentration of electrolytes in the blood. And then things like androgens, such as testosterone and estrogen, will affect sexual development, sexual behavior, and a variety of other reproductive and non-reproductive functions. So they have different functions as far as what they do as hormones, but generally they're going to enter into the cell nucleus and bind, altering gene expression. Another type of lipid-based hormone is vitamin B hormones. These are all, vitamin D hormones. These are derived from vitamin D. They also, like the steroid counterparts, because they're lipids, they bind into the nucleus in a nuclear receptor. So they're internal. And many of these are obtained from food or from the photolysis of the dehydroxycholesterol if you have sun exposure, right? So you can make vitamin D in your skin using UV light. An example is this calcitrol, which is the active form, and it affects transcription of genes that regulate calcium. And the balance of calcium deposition and removal from bone. So this is the relation of vitamin D and bone health. That's why inadequate dietary vitamin D can lead to defects in the synthesis of calcitrol, and it can result in serious diseases such as rickets, in which bones are weak and malformed. There's also retinoid hormones. Again, they're lipid-based and they bind to the nuclear receptor. These are derived from vitamin A, which is derived from beta carotene, and have at least one form of a retinoid receptor. The hormone receptor complexes regulate genes governing cell growth and differentiation generally. They're most active in cells that are experiencing rapid growth. Often you see retinoid receptors upregulated during growth and development of embryos, for example, but also in just cells that undergo rapid growth normally. These are like lung epithelial and skin and immune systems. These are all types of cells that are actively growing and reproducing. And so they're usually utilizing some type of retinoid receptor hormone that tells them to grow and reproduce. So excessive vitamin A can actually cause birth defects because of its relation to this regulation of growth. Another type of hormone we see often talked about are thyroid-based hormones. These are just named because of the place in which they are produced. They act through nuclear receptors, so they are lipid-based, and they act to stimulate energy-yielding metabolism. This is especially in the liver and muscle, and they do this by increasing genes that are key to encoding catabolic enzymes. So these enzymes are lipid-like enzymes that are related to metabolism. Their backbones are also related to the formation of tyrosine. So they have a lipid-like function, but are made from tyrosine or amino acid derived backbones. And you can see, for example, here is the production of one type of thyroid hormone, T3, which is triiodothyronine, T3, which goes through these steps of conversion from thyroglobulin, tyrosine, all the way down to T3. Now notice that these molecules utilize iodine as part of their structure. T3 having 3 iodines and T4 having 4 iodines. This use of iodine in the production of thyroid hormones is key to their production, but is the reason why things like thyroid cancer can occur if you have iodine, which is radioactive, for example. And radioactive iodine can actually be used, supplemented to be used, to then enter and kill the cancerous cells within the thyroid. So the intake of iodine, usually from iodinized salt, for example, or other nutrients, is important for thyroid health, and malfunctions in iodine intake can lead to poor thyroid health. Another type of hormone that we saw actually in our chapter about nucleic acids, amino acids, is nitric acid, which is actually a free radical signaling molecule that's made from arginine. So it's made from an amino acid and from oxygen by the enzyme nitric oxide synthase. This is an enzyme that acts very close to its point of release. So it's a paracrine type hormone, and it enters the cell and activates glucinyl cyclase, ultimately increasing cyclic GMP. Cyclic GMP in the intracellular receptor. This is a secondary molecule which can then activate protein kinases, which lead to the relaxation of contracted proteins in smooth muscle and blood vessels. So it lowers your blood pressure. Now, this is why nitric oxide can be utilized as laughing gas in dental offices because of its relation to relaxing proteins and smooth muscle and lowering blood pressure. Now, when talking about hormonal signaling, it's important to classify two main types of ways that hormones can signal through the body. One is called top-down. This is where signals originate in the brain, and then the signal is sent out to the body. These include hormonal signals such as oxytocin, vasopressin, or cortisol. Again, all originating in the brain and then sent out for signaling. Bottom up is when a signal originates from elsewhere in the body and then can send a signal or a message to the brain. This includes epinephrine or adrenaline, insulin, and leptin. These all are going to be produced in other organs, but can send messages to the brain about the status of the body, essentially. So again, we have our top down and bottom up. So some of the main endocrine glands that are related to hormonal signaling, of course, include the brain itself, in particular in the areas of the hypothalamus and the pituitary, which the hypothalamus is the coordination center of the endocrine system. Ultimately, it is kind of like the central station. And the pituitary has two functions, the anterior and posterior, both functioning as distinct parts. There's the thyroid and parathyroid adipose tissue. Fat tissue actually is important as an endocrine gland itself in signaling. Your adrenals, which are on top of your kidneys, pancreas, ovaries, and testes. These are all very important to the endocrine as endocrine glands themselves for signaling. So this is just an example of some hormones and their target tissues, starting with the neuroendocrine origins of signals, going down into their ultimate targets. For example, the central nervous system can first talk through the hypothalamus by releasing hypothalamic hormones. These can then signal to a variety of things. For example, they could be somotropin, which is a growth hormone, which signals to the liver and bone, being its ultimate target. Or you could have thyrotropin, which signals to the thyroid. This is a secondary target. The thyroid can then release its own hormones, for example, thyrodoxine, triptonine, or T3 or T4 or T3, these thyroid hormones, which then will signal to things like muscle and liver. Again, the hypothalamus itself is the coordination center of all of the endocrine system. It's a very small region in the forebrain in animals with skulls, and it receives and integrates nerval signals from the central nervous system. It synthesizes only small peptide hormones, oxytocin and vasopressin. And it also synthesizes several factors that regulate the function of the anterior pituitary gland, also a part of the brain, which is responsible for signaling. The pituitary gland can then release hormones that target other glands. For example, the posterior pituitary gland contains the ends of axons from the hypothalamus, produces short peptide hormones, again, that can signal this portion. The anterior pituitary, it actually receives releasing factors from the hypothalamus via the blood vessels and produces long peptide hormones such as tropins. So these both can talk to the hypothalamus in different ways. Again, posterior is actually being relayed information directly from axons from the hypothalamus, whereas the anterior pituitary is the ultimate endocrine organ that's receiving information from the blood vessels, right? And then can release information back into the blood vessels to then travel to other areas. So let's talk about those two main hormones that are released from our pituitary glands, which is oxytocin and vasopressin. Oxytocin is going to promote contraction of smooth muscle. It's one of the ones responsible for contraction during the uterus and labor. Milk release from mammary glands is another connected hormonal response to oxytocin. Vasopressin can promote things like water reabsorption in kidneys to maintain salt balance, or constriction of blood vessels to increase blood pressure. Both hormones play roles in social behavior too, such as pair bonding. And these central nervous system effects do not involve the pituitary secretion. So here is the hormone production and release that occurs following the intake of information into the central nervous system and how it kind of is cascaded through to the other glands. This is the cortisol cascade from the hypothalamus. Things like fear, infection, and so on actually send electrical signals to the hypothalamus in the brain. And this is going to result in the release of the corticotropin releasing hormone from the hypothalamus itself, which then activates the anterior pituitary to release the corticotropin molecule, which then signals to the adrenal glands to release cortisol. Cortisol is the end product and it feeds back, and it can actually inhibit the tissues prior, the interior pituitary and the hypothalamus itself, from continuing to activate the adrenal glands. Cortisol, of course, can also be released into the blood to signal to the muscle, liver, and adipose tissues themselves. When cortisol reaches these areas, it can act on the liver, fat, and muscle, for example, to promote fatty acid and glucose release in response to pain. It changes the types of enzymes rather than activating existing enzymes. So in fat, it increases fatty acid and glycerol release. In liver, it stimulates gluconeogenesis. And in muscle, it breaks down proteins to export amino acids to the liver and gluconeogenesis. Generally, what you're seeing is that the cortisol is signaling to all of these areas to get energy, right? It's breaking down proteins, exporting them to the liver. It's increasing fatty acid and glycerol release. It's making more blood glucose through gluconeogenesis. This is all so that you can have that fight or flight response, right, with cortisol. So generally, this molecule is going to be around when you are stressed, right? It's going to increase the amount of blood glucose available, increase fatty acids available, and break down proteins. These are all stress responses. Generally, cortisol counteracts the effects of insulin, which is important when we're going to be talking about insulin and its impacts on the body. Remember that insulin is actually going to attempt to lower blood glucose, whereas cortisol is increasing the amount of glucose available in the blood. Continued release of cortisol in the stress release can lead to something called adrenal fatigue, where the effects of cortisol are ultimately lessened. You're so used to so much cortisol release that you now need even more to have an impact on you. Cortisol release can essentially become compromised, and this can damage bone, muscle, immune system, and other hormone functions because of the extreme amount of cortisol that is now needing to be released to have impact. All right, well, that is the end of our first section on hormonal regulation.